Hi everyone, this is chapter 12 on the nervous system and nervous tissue. So as always, we have our learning objectives. So let's start talking about the two main divisions of our nervous system. So we have two regions. We have the central nervous system and the peripheral nervous system. Your central nervous system, also known as the CNS, is the brain and the spinal cord, and the peripheral nervous system, the PNS, is everything else. There are two ways to consider how the nervous system is divided functionally as well. The basic function of the nervous system is sensation, integration, and response. The control of the body can be through somatic or autonomic systems that are largely defined by the structures that are involved in the response. Looking at the nervous system functionally, the first component is the idea of sensation. And what sensation is receiving information about the environment to gain input about what's happening outside of the body, or sometimes what's happening inside the body. So it's receiving information about the world around and within you. The sensory function can register the presence of a change from homeostasis or a particular event in the environment known as the stimulus. The second component is once that signal is received, it's transmitted to the central nervous system where it's handled by a special center that's going to analyze that data and make a decision about it. In this example, the decision is whether or not we're going to turn on the hot water faucet. So we're picking up information about the temperature of the water. That would be the first component. And then we're making a decision once that information is brought into the brain of do we turn the hot water on? Is the water warm enough? The third component is called the motor output. This is what carries the signal to the skeletal muscle to perform the action based on the sensory input and the integration centers. And in this picture, the physical process of opening the faucet because we decided that it was too cold. So looking at these divisions of the nervous system, I think a flowchart really helps with this. So overall, we're talking about the nervous system, which is the last system we cover in this particular course. We can divide that into the central nervous system and the peripheral nervous system. And when we talk about the central nervous system, we're talking about the brain and the spinal cord. When we talk about the peripheral nervous system, we can subdivide it into the autonomic nervous system and the somatic nervous system. The somatic nervous system is your muscles. That's what we learned in the last chapter. The autonomic nervous system is going to be subdivided even further into the sympathetic and the parasympathetic nervous systems. Nervous tissue is present in both your central and your peripheral nervous system, and there's two basic types of cells that we talk about, neurons and glial cells. A glial cell is one of a variety of cells that provides a framework of tissue that supports the neurons and their activities. We sometimes refer to glial cells as helper cells or nurse cells because they help the neurons do their job. The neuron is the one that's carrying the communication function of the nervous system. So it's the one that's transmitting signals. The glial cell is just helping it do that. So let's talk for a few minutes about the structure and the anatomy of our neuron. So these are the ones, again, that are responsible for conducting electrical signals. They're going to communicate information about sensation and produce movement in response to stimulus, as well as all of the things that we associate with our mind when we're talking about thinking and cognitive processes and learning and all those sort of things. So here we have a neuron over on the right-hand side. So let's talk about the parts. The soma is the body, so the gold colored structure you see there. All right, that's where you see your nucleus, which is the purple spot in the middle of that, and our major organelles are located. So we're going to see all of these organelles that we've seen in other cells. We're going to see them in a neuron as well. The dendrites are the extensions from that cell body that receive information. It's what looks like the branches of a tree. All right, so those are your dendrites, and they're going to be receiving information from other neurons, and there is a little teeny tiny space between the neuron in front of it, which is what they're showing you there, and the actual dendrite itself, and that space is going to be called the synapse. The axon is the long tail-like structure that comes from the cell body, projects down to the next cell in the series. So if you think about neurons, there's not just one great big long neuron. It's like a whole bunch of individual neurons, one right after another. So you've got the axon of one that's going to be in contact with the dendrites of the next one. And then you're going to have the dendrites of that, or the axon of that one, in contact with the dendrites of the next one. So one right after another, like telephone poles when you're driving down the highway. The axon hillock is the enlarged funnel-shaped space that the axon emerges from the cell body, and the axon terminal is on the other end, and that's where the axon meets the next cell in the series. So the structural classification of neurons is going to be based on the arrangement of the dendrites, the cell body, and the axons. So there are several different ways for neurons to exist. We've been looking at your standard multipolar neuron, but not all neurons look like that. So we're going to look at them based on where they can be found. We can also look at what chemicals they use to communicate with each other. There's a lot of ways that we can classify our different neurons. We'll talk about this idea of the arrangement of the cell body and the neuron, the dendrites and the axon, and the different types of neurons we see there. So the one that we've been looking at already, and the one most people are familiar with, is what's called a multipolar neuron. So you have one axon and at least two dendrites, but you see that there's a whole lot more dendrites there, and that the cell body's right there in the middle of all of the dendrites. This is the most common type of neuron, and they are found both in the central and the peripheral nervous system. Another way to be a neuron is called a unipolar neuron. These only have one process that emerges from the cell body itself, and you can see the cell body is off to the side. And then you have that one little short process, and then it kind of divides and goes both directions. So the cells have an axon that emerges from the cell body, but it splits. So you can see that in the picture. And at one end you have the dendrites, and at the other end you have the synaptic connections to the next target. All unipolar neurons are sensory, so they're incoming information about the world around you. And then finally, we have bipolar neurons. These have two processes with the cell body smack dab in the middle. So notice how it's not over to the side, it's actually in the middle there. Bipolar neurons are not common. They are found mostly in the olfactory epithelium, where the sense of smell is sensed and information is conveyed about that. And they are also part of the retina. So those are our different kinds of neurons. Now let's talk about glial cells. Remember, those are the helper cells. So these are the support cells, is another way to say it. These ones do not conduct electrical signals that we'll be talking about later, but are instead there to help the cells that do, which are the neurons. There's 4 types of glial cells in the central nervous system, and there are 2 types of glial cells in the peripheral nervous system. So we're going to talk about the central nervous system first, and we're going to talk about astrocytes and oligodendrocytes, microglia and appendable cells, and then we're going to look at the peripheral nervous system and look at satellite and Schwann cells. In this little chart is showing you what the basic function of those are, and you can see that the astrocyte and the satellite cell basically have the same function. It's just found in a different location, one being in the central, one being in the peripheral. And the same thing with the oligodendrocytes and Schwann cells. They basically do the same thing. It's just a difference in where that cell is located. So let's talk about the central nervous system cells first, and we will start with talking about an astrocyte. So astrocytes are called astrocytes because they are shaped like a star. They provide critical support, metabolic and functional support to neurons. They are the most numerous cell type in the brain. There are 10 of these for every 1 neuron, so there's a lot more of these. They maintain homeostasis. They help to regulate the blood-brain barrier that we'll be talking about. They supply nutrients, and they help to modulate the synaptic activity. So the astrocyte is the blue one that you see there. All right, another type of glial cell in the central nervous system are the oligodendrocytes, and their main function is to create the myelin sheath. So the myelin sheath is a fatty insulation that's found around the nerve axons that helps to speed up the electrical signal, what we call the nerve impulse. But they also provide metabolic support to the axon, and they help maintain the stable environment that's needed for proper neural function. And that proper neural function extends to things like neuroplasticity, which is how the brain rewires and repair. So looking at this picture, we see our gold neuron, and then you see that oligodendrocyte in blue. And notice that it takes its little processes, its little arms, and it wraps them around the myelin sheath. So it makes these individual little wrapped up sections of this fatty insulator. So one oligodendrocyte could do this with several different neurons. The little space between each of the nodes of myelin sheath is called the node of Ranvier. Another type that we talk about are the microglia, often called the guardians of the brain. These are the smallest of the glial cells, which is why they're called micro, and they act as immune sentinels. Their jobs are to serve as debris cleaners, and they use the process of phagocytosis to remove dead cells and pathogens and any other unwanted substances from the brain. So you can see your purple microglial cell on there and all of the things that it goes after to keep things nice and tidy inside of the cell, inside of the brain, sorry. The final type of glial cell that we talk about in the central nervous system are going to be the ependymal cells. These are specialized ciliated glial cells. Typically, they are simple cuboidal cells that form a single layer of epithelial tissue that lines the brain's ventricles. So inside of your brain, there is a some hollow rooms or some hollow spaces that are called the ventricles, and the spinal cord also has an empty space in it as well, called the central canal. And so what's lining those areas are these ependymal cells. They are crucial for secreting and circulating cerebral spinal fluid via the cilia that are attached to the apical surface of those cells. They also regulate the cerebral spinal fluid brain nutrient exchange, and they clear any metabolic waste. So that allows us to move over to the two types of cells that we see in the peripheral nervous system that are considered glial cells. The satellite cells, which are sensory and found in sensory and autonomic ganglia, where they surround the cell bodies of the neurons. These guys are going to provide support, they perform similar functions in the periphery as the astrocytes do in the central nervous system. Satellite cells are probably the least understood of the glial cells. When I was in college taking this course 40 years ago, we didn't even know about satellite cells. So this is information that's come about in the last few decades. Schwann cells are going to insulate the axons with myelin in the periphery in a very similar way that the oligodendrocytes, but there's a couple of significant differences. So here, the gold is a ganglion. It's a whole group of cells clustered together, cell bodies clustered together, and the satellite cells, you can see those kind of plastered all around the outside of it. They're the red cells, and so again, they're similar to what the astrocytes are doing. I've talked a little bit about myelin, and let's talk about it a little bit more. Myelin is used to create a myelin sheath. It is a lipid-rich substance that surrounds the axon. It protects and insulates some axons and is absent in what they call non-myelinated axons. So when we talk about myelination of an axon, not all axons are myelinated. Only some of them are. It's been estimated that if every single axon in your spinal cord was myelinated, your spinal cord would be as big around as a barrel. So you can imagine that would be not a very easy way to move around. So it's only going to be found on axons where we want the speediest transmission. So again, how that happens is you've got the axon in the middle, and in this case, you've got a Schwann cell. A Schwann cell is going to take its whole body and wrap it around that little section of axon. we saw something a little bit different when we looked at the oligodendrocyte. When we looked at the oligodendrocyte, it was just taking one of its arms and wrapping it around a little piece of the axon, and it was taking another arm and wrapping it around a different piece of the axon, or a completely different axon on a different cell. In the case of our Schwann cell, their whole body becomes the node of myelin, and you can see the nucleus is actually in the node. So again, it is a protein and phospholipid mixture that forms an insulating sheath around nerve fibers and increases the transmission of the electrical signal along the axon. Each node of myelin in the peripheral nervous system is made of a single Schwann cell that wraps itself entirely around the axon of the neuron, and again, the space between those is called the node of Ranvier. So those are your different types of glial cells that you'll want to know about, and make sure that you can explain each of them. Gray matter versus white matter, because the way the neuron cell bodies and the axons are arranged, we have two distinct colors or regions that we see. Whenever you see gray matter in the brain, that is cell bodies and dendrites and unmyelinated axons. When you see white matter, it is mainly myelinated axons. So that's the difference between those two. Some other terms to know: the difference between nuclei and ganglia. So nuclei is kind of confusing because we were talking about the nucleus of a cell. But when we're talking about in the cell body or in the nervous system, the groups of cell bodies of neurons clumped together can be either called a nuclei or a ganglion. So when we're talking about a cluster of neuron cell bodies in the central nervous system, we call it a nuclei, so in the brain and the cord. When we're talking about a cluster of neuron cell bodies in the peripheral nervous system, we call it a ganglion. So they're the same thing. They're just groups of cell bodies of a neuron. It's just where are they located. So this gets us to talking about membrane potentials, and I talked about this a little bit with the cell membrane, and now we're going to talk about this in a much more robust way and understand how cells, particularly neurons, but muscles do this as well, are going to send electrical signals. So the cell membrane is primarily responsible for regulating what crosses the membrane. You saw that all over chapter 3 when you were learning about how things move from one side of the membrane to the other. Charged particles, which are hydrophilic by nature, cannot pass through the cell membrane without some type of assistance by the transmembrane proteins. And you learned about channel proteins and carrier proteins and all that sort of stuff in a previous chapter. So let's talk a little bit more specifically about some of those channel proteins. They come in 3 different varieties. The first one's called a ligand-gated channel. A ligand-gated channel opens because a signaling molecule, what we call the ligand, is going to bind to the extracellular region on the outside of the cell. So you see we've got, in our picture right here, we have our little integral protein. That's the blue thing that's in our cell membrane right there. And then acetylcholine is going to bind. And when that acetylcholine binds, it opens up that channel. So the little red acetylcholine triangle that you have there, it binds. And when it does that, it opens up the channel and it allows other molecules to move through. So that's what's meant by a ligand-gated channel. A mechanically gated channel is going to open because of physical distortion of the cell membrane. And so this is what we see with the a lot of the sensory type things like your sense of touch. And so this is going to cause a deformation or change in the shape of the receptor, the channel, and allow things to move through. And then finally, you have voltage-gated channels. Voltage-gated channels are going to respond to changes in the electrical properties of the membrane in which they are embedded. So normally the inner portion of the membrane is at a negative voltage. We looked at that before. We looked at the idea the inside of the membrane at the cell membrane is not slightly negative. And when the voltage becomes less negative, the channel begins to open and allow ions to cross. And so that's exactly what you're seeing in this picture. All right, so the membrane potentials. The electrical state of the cell membrane can have several variations. These different variations are called the membrane potential. A potential is a difference of charge across the cell membrane. So there's difference between the inside and the outside. And this is measured in millivolts, which is abbreviated little m, capital V. The standard is to compare the inside of the cell relative to the outside, so the membrane potential is a value that represents the charge on the intracellular side of the membrane on the outside, with the outside being zero, relatively speaking. So we measure this with a little voltmeter, and that's what they're showing you there. So we could take our little teeny tiny probe and put it into our cell membrane. We could measure, and we would see at rest the cell is more negative on the inside than it is the outside, and that's what they're showing you with the plus signs and the negative signs. All right, so the inside of the cell is the cytosol side. The outside of the cell, where the extracellular fluid is the blue side, and that's the positive side. This is a cell at rest. So when our cell is at rest, in other words, it's not transmitting an electrical signal, the ion channels are closed with the exception of leakage channels because those are always open. The ions are distributed across the membrane in a certain way. The concentration of sodium outside of the cell is greater than inside, and the concentration of potassium is greater on the inside than the outside. And what's keeping it this way are those sodium-potassium pumps. When the ions that are distributed across the membrane at these concentrations, the difference in charge is measured at negative 70 millivolts. So when we're at rest, that cell has a charge, if we could measure it with our little voltage meter, of negative 70 millivolts. And that negative is important in front of that. And we call that the resting membrane potential. So let's start this discussion with talking about graded potentials first. So graded potentials are local changes in that little negative 70 millivolt membrane potential we were talking about in just one little piece of the membrane. It's not happening everywhere. It's just in one little place. And this is usually on the dendrites of the neuron. The amount of change in the membrane potential is determined by the size of the stimulus that's coming in from the previous neuron in that one little spot, and that's what's causing it. Graded potentials can be of two sorts. They can be either depolarizing or hyperpolarizing, and for a membrane at resting potential, a graded potential represents a change in that negative 70 millivolts that I just talked about. So if you're at negative 70 and you get a little zap of an electrical signal and it happens to be excitatory, then you go from negative 70 up to negative 50. Or you could receive a signal that is hyperpolarizing, and that means that it's going to go from negative 70 to maybe negative 90. But this is not happening on the entire membrane. It's just happening in one little place where it's in contact with the neuron that is in front of it. So they're just showing you the difference between depolarizing and hyperpolarizing. So I just want to point this out to you that we're looking at the number line over on the left-hand side, and we're looking at time across the bottom. But see, we are at negative 70. That's our resting membrane potential. If we have a depolarizing event, it's going to go from negative 70, start to go up towards negative 60. We're getting less negative. If it is in a hyperpolarizing potential, then we're going from negative 70 down to possibly negative 90. So it's going further away from zero. Depolarizing graded potentials are often a result of sodium ion or calcium entering the cell. Both of these ions are found in higher concentrations outside of the cell than inside, and because they have a positive charge, they will move the cell membrane potential, causing it to become less negative. We're adding positives to that negative 70 and causing it to go up to negative 60. Hyperpolarizing graded potentials can be caused by potassium ion leaving the cell or chloride ion entering the cell. If a positive charge moves out of the cell becomes more negative, and if a negative charge enters the cell, then the same thing happens. So that's a graded potential. I refer to it as a little mini earthquake. It just happens at one little spot and it travels a short distance and then it stops. So it's like a little earthquake that happened out in California. We don't feel it over here, over on the East Coast. We just feel it out in California. So graded potentials are little mini stimulus electrical signals that are coming in all over the dendrites, and they can be hyperpolarizing or depolarizing. Now we're going to talk about an action potential. So an action potential is triggered when there are enough changes in the membrane potential, that -70, that it reaches a critical level known as threshold. And once it hits that threshold point, um, it is going to fire what's called an action potential. If it doesn't reach the threshold point, there's no action potential, only the graded potential that caused that little mini earthquake there. So we have a whole bunch of little mini earthquakes happening all over the dendrites. And if we have enough of them that are excitatory, we're going to trigger what's called an action potential. Action potentials are all or none. They either happen or they do not happen. They are all identical. There is no such thing as a bigger action potential. They're all the same, exactly the same, and they don't decay over time and distance. So it'd be like if we had an earthquake out in California and we could feel it at the same intensity all the way over here in South Carolina. So here is what our action potential looks like. So you notice that we're at -70 on the left-hand side on our membrane potential there. And so we have depolarizing events. We have these little mini little bursts of electrical activity that are coming in all over those different dendrites. And we get enough of those that we start to reach what's called threshold, which is -55 millivolts. And when that happens, we're going to have a massive depolarization event. Notice that our membrane potential goes up to +30, and then it's going to start to repolarize, and it's going to drop back down. In fact, it's going to drop down below -70 in a hyperpolarization event, and then it's going to level back off at -70. That's a signal. That was an electrical signal that just traveled down that axon, and that's what it looks like. So when we're looking at the little graded potentials, those are happening up on the dendrites. If we get enough of those, we can cause an electrical signal to pass across the cell body and travel down the axon to the next cell in the series. It's going to be received at the next cell in the series as a graded potential. And if we get enough graded potentials coming from enough different neurons on the next cell in the series, we could have that pass on, that action potential pass down to the axon and pass to the next neuron. So just a little bit more detail, that's what happened at -70. We're said to be at rest. We get a stimulus. We get enough stimulus that we reach threshold at -55. The voltage rises till we hit about +30, and then it starts to fall. And as it falls, it actually goes beyond the -70 to -80 at -90 or so, and then it returns back to rest. Once the signal arrives, the sodium channels are going to open, and sodium is literally going to rush into the cell. The resting membrane potential, again, is -70 millivolts. So as sodium, which is a positive ion, enters the cell, it causes the -70 to start rising towards zero. That's known as depolarization, meaning that the membrane potential is moving towards zero. So depolarization means that we're going from -70 up towards zero. As the membrane potential reaches +30, other voltage gated channels are going to open. Remember, the ones that are going to open in response to electrical signals. And that's going to include things like the potassium ion being able to enter or leave. So as the potassium ion leaves the cell, the membrane potential moves back now towards resting. That's called repolarization. That means that the membrane voltage is moving back towards -70 at the resting membrane level. Potassium ions are going to reach equilibrium when the membrane voltage is below -70. So we have that little brief dip below down to -90 that I referred to as hyperpolarization. That's going to occur when the potassium channels are open. Those potassium channels are slightly delayed in their closing, which is accounting for that dipping down to -90 and then rising back up to -70. They're just a slower channel. So as that as an action potential travels down the length of the axon, the action potential is propagated because more voltage gated sodium channels are opened as the depolarization spreads down. I always liken an action potential moving down the axon like a whole string of dominoes. Starting at the very base of the tail, you just have one domino knocked down, one right after another, all the way down to the very end. And so that's the idea of what propagates, and that's all because of sodium ions that are entering into the channels as it moves down. So as the sodium moves a short distance along the cell membrane, its positive charge is depolarized at a little piece of the membrane, one right after another, just like dominoes in a row. And as that depolarization spreads, new voltage gated channels are opened and more ions of sodium are going to rush in, and it's going to cause the depolarization to happen in the next little section. So showing you an idea of how that's happening. We're just conducting it in what's called saltatory conduction. So I mentioned the idea of an action potential moving down the axon, and if the axon is unmyelinated, it moves just like that string of dominoes. But if the axon is myelinated, what's going to happen is that electrical signal is literally going to skip from one node of Ranvier to the next. So the node of Ranvier is that little bare spot where there's no myelin. And so it literally is going to hop and move from bare spot to bare spot. It's called saltatory conduction, and saltatory means to skip. So I want you to think about it like this. If you were to walk from one side of the room to the other, it would take you a certain amount of time to do that. But if you skipped, it would be a lot faster. And that's the whole idea of the myelin sheath. Remember, I mentioned that the myelin sheath is in place to increase the conduction speed of the axon, of the action potential down the axon. And so that's exactly what's happening. How we're increasing that is that we have the that are acting as insulators, so that electrical signal literally has to pass over and hop to the next one. So you kind of see that idea of how it just moves down. It just hops down the axon. On the left-hand side, you have one that's not myelinated. So you see that it doesn't travel as easily. When you myelinate is, notice how fast it travels from the cell body down to the terminal. So a postsynaptic potential, a PSP, is the graded potential on the dendrites, as I mentioned earlier, of a neuron that is being received from other cells. So a neuron is receiving information at its dendrites from a lot of different cells, thousands of different cells at the same time. Postsynaptic potentials that are coming in can be either excitatory or inhibitory. We refer to them as EPSP if it's an excitatory postsynaptic potential, or an IPSP if we're talking about it being an inhibitory postsynaptic potential. So as all of these different signals are coming in, so let's say we're at the dendrites and we're getting 500 excitatory signals coming in, and we're also getting 450 inhibitory. If we get more excitatory signals coming in than we do inhibitory signals coming in, then an action potential is generated, and that signal is passed down the axon to the next neuron. It's going to be received at the next neuron as a graded potential. So the job of the axon hillock, that place where the axon comes out of the neuron, the job of the hillock is to actually be the accountant. It's going to sum how many excitatory signals came in compared to how many inhibitory signals came in. If enough excitatory came in, then it's going to pass that action potential, start generating action potential, and run it down the axon. If more inhibitory come in than excitatory, then it's not going to generate one. All right, so that's the whole idea about these graded potentials that are coming in. They're determining whether or not an action potential is actually going to occur. So let's talk about the synapses for a few minutes. There's two types of connections. Remember, the synapse is the space between two active cells. So it could be between a neuron and a neuron, or a neuron and a muscle cell, as we saw from the last chapter. If it's called a chemical synapse, there is a chemical that's going to jump from one, or diffuse over from one cell to the other. We saw that in our muscles with acetylcholine. We saw acetylcholine released by the axon uh, of the neuron onto this chemical space, or this synapse, and then it drifted over and bound to the muscle side and generated the action potential over there. So that's a chemical synapse, where a chemical, so it's electrical, chemical, electrical. So a chemical is responsible for mediating that space between the two. We can also have an electrical synapse. This is a direct connection between two cells so that the ions pass directly from one cell to the next. And if one cell is depolarized, that joining cell will also be depolarized. Electrical synapses are much less common. There are very few of them. And the reason that they're not particularly common is that a chemical synapse is going to prevent the electrical signal from going backwards. In electrical synapse, you have a direct connection, and the chemical signal could actually go backwards. And we don't want a electrical signal to go back up the axon the way it came down. So electrical synapses are only found in a couple little small areas of the brain. They're more common in newborns while their brain is forming. So let's talk a little bit about those neurotransmitters that we have in chemical synapses. So we have several different systems or groups of neurotransmitters. One is called a cholinergic group or system, and this one is all about acetylcholine. So acetylcholine is the name of the neurotransmitter. The receptor that it binds to can either be a nicotinic receptor that's found like at the neuromuscular junction, and it's found in other synapses as well, or a muscarinic receptor, which is an acetylcholine receptor. So one thing that I want to point out here is that we actually have a receptor in our body for nicotine, which is one of the reasons why cigarettes are so addicting, because we actually have a receptor in place already. And that makes it really hard for people to stop smoking because of the nicotine. It was no accident that when they made cigarettes that they used nicotine in them. If they'd made them without nicotine, they wouldn't have been addicting. Another group or system we have is called the amino acids. This includes glutamate, GABA, and glycine. Some other groups of neurotransmitters. One is called biogenic amines. This is a group of neurotransmitters that are enzymatically made from amino acids. When we use the term neuropeptide, this is a neurotransmitter molecule that has a chain of amino acids. It's made of a chain of amino acids that are connected by peptide bonds. So those are the different big categories we have of neurotransmitters. There are approximately 50 different chemicals that act as neurotransmitters. Some of them act as a neurotransmitter in the brain, and they act as a different kind of chemical someplace else in the body. What are neurotransmitters? What's their effect? So the effect is on the postsynaptic cell, or element as they refer to it, and it is entirely dependent on the type of receptor that it binds to. So if there is no receptor on the membrane on the postsynaptic cell, the cell that is after the synapse, then the neurotransmitter simply has no effect. And that's how all receptors work. If there is no parking spot for that neurotransmitter or that chemical on the cell, then that chemical or that neurotransmitter in this case just simply floats on by. It's like if you go to the movie theater and there's no parking spots in the movie theater, you're not going in the movie theater, are you? Right? And so that's the idea of what a receptor does. It's literally a parking spot for a chemical on the postsynaptic cell. And as long as it's there, then that chemical can have an effect on that cell. And if it's not, it can't. Okay, so the depolarizing or hyperpolarizing effect is also dependent on that receptor. So when acetylcholine, for example, binds to a nicotinic receptor, the postsynaptic cell becomes depolarized and the effect is excitatory. When acetylcholine, the same molecule we're talking here, binds to a muscarinic a receptor on that postsynaptic cell, it could be either excitatory or inhibitory. So it depends on the type of neurotransmitter, but mostly it depends on the receptor. So the effect that you have on the postsynaptic cell is dependent upon the type of receptor.